I really enjoy being outdoors and exploring the environment. As a kid, we would go as a family to an island called North Captiva. It was super cool to just be able to swim around and find sand dollars or see dolphins and sharks fighting. And I think that just sparked something so that I wanted to know more about why those organisms live there and what other organisms live there. This project we refer to as the Sanctuaries MBON project. MBON is an acronym for Marine Biodiversity Observation Network. The aim for this project is to be able to use environmental DNA as a signature for different organisms within the water column so that you can use that as a monitoring tool. Environmental DNA, or eDNA, is the skin cells or scales extracellular DNA that has been suspended in the water column. So basically any organism that's swimming through the water sloughs off their skin or scales, and that then becomes incorporated into the water. And over time those skin cells will decompose, and that allows for us to have extracellular DNA, or just DNA that's floating in the water. We use that DNA to then be able to associate that DNA to a certain organism or a certain species.
In the last few years, the ability to sequence has gotten much better.
Right.
The costs have gone down. Here at MBARI, we're working on the ability of using autonomous vehicles to collect the samples.
Right.
And so we can imagine how someday we'll be in a place where we can do this in a much more routine fashion.
We're examining changes in biodiversity over time, as well as examining biodiversity that maybe we haven't been able to capture before.
I got interested in it in terms of really trying to understand how to interpret the eDNA, how many fish had to have been around. For example, if you measure a certain amount of their DNA in the water. You guys collected the eDNA samples alongside the rockfish trawling, so that's going to be really interesting, I think. We have 300 samples from that cruise.
Yeah, and there were a lot of trawls that happened on that cruise, and being able to compare and overlap answers from what the traditional methods assessed versus what we'll be able to assess through eDNA, I think will be really important. This is a large collaboration with multiple different teams.
We have this other contingent over in the Florida Keys National Marine Sanctuary.
Right.
So they're doing basically the same types of measurements that we are for eDNA, and so it's going to be a really interesting contrast, right? The rich, cold, and productive Monterey Bay against the warmer, clearer, tropical Florida Keys.
Working with the National Marine Sanctuaries, I think, has also been really important, because we're able to then inform them of these methods and how they might be able to use that in the future for monitoring this important marine sanctuary. It's nice to be able to work on projects where not only are you passionate about it, but you also recognize that there is application and implementation of what you're discovering. Managers as well as decision makers may be able to use this type of information to be able to make more informed decisions, to be able to protect those marine environments as well as be able to regulate those marine environments better.